Today I'll be presenting a poster about the potential use of Raman spectroscopy for trait identification in peanut. This is Jaime Shumaker with the Texas A&M AgriLife Research Center in Stephenville, Texas, along with J.M. Kaysen, C.E. Simpson, D. Karowski, and M.D. Burrow. In the past, trait identification of peanut germplasm for breeding was performed by expensive, time and labor intensive, and in some cases destructive, hand sampling techniques, as well as traditional field trials and hand selections. These constraints often lead to underrepresentative trait identification due to limited sampling and limited time, as well as the introduction of possible human errors. There's a significant need for time-saving, non-destructive, and non-invasive methods to identify traits in peanut breeding lines. With the introduction of Raman spectroscopy, these hurdles may be overcome. In the spring and summer of 2020, the Texas A&M AgriLife Peanut Breeding Program in Stephenville, Texas, purchased a handheld Raman spectrometer and initiated a preliminary evaluation of Raman spectroscopy for use in identity preservation and peanut trait identification. In preliminary studies, Raman spectroscopy was successful in identifying wild peanut accessions in both cultivated and wild peanut seed with up to 100% accuracy. In other research, an imposed drought trait study was conducted where 2 cultivated peanut varieties were grown to maturity in a greenhouse and then subjected to one of 3 treatments: well-watered, drought stress, and high salinity stress. Raman spectroscopy was used to sample leaflets for the potential to identify drought stress and salt stress before it is visible with the human eye. Plants were arranged in a complete randomized block. Sampling began on day 1 of treatment and continued for 5 days. By the end of day 5, plants were no longer viable for sampling due to induced plant decline caused by water treatments. Raman spectra were collected each day with a handheld Resolve Agilent spectrometer equipped with an 830 nanometer laser source. Data for both projects was subjected to partial least squares discriminant analysis. Based on the data collected during our 2020 studies, we believe Raman spectroscopy has the potential to be a valuable tool in field applications for breeding and evaluation of wild and cultivated peanuts. Preliminary results show that Raman spectroscopy can be used to identify cultivars and accessions with high accuracy. In addition, plants under drought stress and high saline soil conditions can be identified before the stress is visible with the human eye. We feel Raman spectroscopy has possibilities in identity preservation and identity identification of both biotic and abiotic stressors. I'd like to thank our funding agencies, Texas A&M AgriLife Research, Texas Peanut Producer Board, and the National Peanut Board for their continued support. Thank you.